Hi guys, sorry I forgot to unmute myself. We are here to talk about Hardy-Weinberg. And so there are 5 I can statements here. And the first one is, I can explain how variation in a gene pool affects the stability of a population. All right, so what I want you to picture is well, let's start with one population. Okay, so parallel universes. Okay, in the first universe, one population of deer, we'll go with, and they're all brown. They're all the exact same color brown. They live in a brown forest. Everybody's happy, everybody's doing fine. The ones that are killed and eaten are being killed and eaten due to other traits like speed or how loud they are or something like that, but not color. Okay, so then let's say this is kind of a dry forest and that's why it's brown. Well, then the weather pattern changes and it starts to get a lot of rain and it turns really lush and green. Okay, so now you have these brown deer in this green environment, and so it's not a good mix, it's not a good fit anymore. And so what happens is we start to see those brown deer get eaten because they're standing out. Everybody can see them. Their predators can see them. And so they get eaten and the population dwindles down to zero. Okay, unless some of them are really good at hiding or something, but let's just say they're not. And so population, because they're all the same, they can't adapt to this new green environment and they all die. Okay, so that's in that universe. Now let's go to this parallel universe. In this scenario, we have the same population of deer, or the same kind of deer, not same population, same kind of deer, but some of them have a mutation that allows them to have a relationship with algae. So algae live in their fur and make them kind of a greenish brown, okay? So you have some deer that are just brown, and you have some deer that have the brown fur, but they have some algae in their fur, and so now they're kind of a greenish brown. And same thing, dry forest, it's brown. The green ones don't do so well. They kind of die out, but there's still some there because it's a recessive mutation. So it kind of keeps popping up to be able to work with the algae. And then the weather pattern changes. The forest gets greener. And now the greener brownish deer are blending in better and the brown ones are not. Brown ones get eaten. Greenish ones can kind of camouflage so they don't. And so what you see is the green ones get more prevalent, more common, and the brown ones start to disappear. But what you don't see is the entire population of deer get wiped out because there was variation. Because there was variation, some were able to adapt, some were not. But because some were able to adapt, the population or the species gets to survive even though every individual doesn't. And that is the benefit of genetic variation. It is that it allows species to survive or even populations to survive because when there's a lot of variation, and with sexual reproduction there's a greater chance that when there are environmental changes or other things going on, something will be able to adapt, some member of the population, and therefore survive and reproduce and pass those genes down. All right, the rest of them really have to do with Hardy-Weinberg. So the first, I'm actually going to jump to the third one. It says, I can describe Hardy-Weinberg equilibrium. I'm going to share my screen so you can see what I'm looking at. Not that I'm looking at anything right now. Hardy-Weinberg equilibrium is the opposite of evolution. So basically, I can't talk and write at the same time apparently. That's an ILB equilibrium. Is when there is no evolution occurring. Okay, no evolution. Okay, so if a population is in Hardy-Weinberg equilibrium, it is not evolving. Okay, and if you'll remember, when we have, think back to your 5 fingers, right? When we have evolution, we have genetic drift. So when we're in Hardy-Weinberg equilibrium, we're going to have no genetic drift, which means probably we have large populations. And so those random changes aren't going to make a big effect because each individual is such a small percentage. Second finger, remember sexual selection or non-random mating. So if it's not non-random mating, not non is 2 negatives, that the opposite of that would be random mating. Okay. Then the middle finger is mutations. So if there is no mutations, then we are in Hardy-Weinberg equilibrium. Okay, first finger is gene flow, no gene flow. Okay, and then the thumb is natural selection. So if we're in Hardy-Weinberg equilibrium, there is no natural selection. Okay, so basically if it's in Hardy-Weinberg equilibrium, It is not evolving. And if it's not evolving, that means none of the 5 mechanisms are happening, right? So if it were evolution, we would see genetic drift. We would see non-random mating. We would see mutations in gene flow and natural selection. But when there's no evolution and it's in Hardy-Weinberg equilibrium, we're not seeing any of that. There is none of that, okay? So that is Hardy-Weinberg equilibrium. That's what it is. It's when a population is not evolving. Now, how do we quantify that? How do we know that? What's based on allele frequencies. So if I go to a particular population, and let's say the dominant allele, which we use P in Hardy-Weinberg equilibrium, is 80%, and the recessive allele, which we represent with Q in Hardy-Weinberg equilibrium, is 20%, and that's in 1950. And then we come back in 2050, 100 years later, and we see that these numbers are the same, then it's not evolving. It's in Hardy-Weinberg equilibrium. Okay, so that's how we measure evolution. Do these numbers change? It's evolving. Do they not change? It's in Hardy-Weinberg equilibrium. Okay, so the other 3 have to do with calculations and using the Hardy-Weinberg equation, which is based off of the Hardy-Weinberg equilibrium, or it allows you to see if that's happening or not. Okay. So it says I can calculate allele frequencies and show them graphically. I can explain how Hardy-Weinberg equilibrium is used to quantify evolution. Okay, sorry, I just wanted to make sure this was recording. I don't trust myself sometimes. And then I can use evidence or data to determine whether or not a population is evolving. So let's start with I can calculate allele frequencies. First of all, I'm going to show you the creature that we're looking at. So we're going to be talking about capybaras. They're the largest rodent in the world. Aren't they cute? Look at their little face. Oh my goodness, I could just squeeze it. Precious. It probably would bite my hand off. But anyway, I don't know. They might be nice. I don't know anything about capybaras other than they're really large rodents and they swim. Okay, so let's talk a little about capybaras. So capybaras have webbed toes. Okay. And they have webbed toes because they swim, because they are in the water a lot. Now, they're not water creatures. They're not fish, right? They're mammals. They live on land. I mean, there are mammals in the ocean, dolphins and such, but they are land mammals that go into the water to catch prey and stuff like that. And let's say that in this particular, and this is true, okay, this is a true statement. This next part is what I'm kind of making up for our scenario. Let's say that there's a dominant trait for normal webbing. Okay, and so their little toes, they have, I don't know what their feet, I'm probably going to draw a duck foot here. Y'all know my drawing. Well, that might be the extra webbing. Let's do this. Okay, so let's say there's a little bit of webbing. Let's say their toe bones kind of go down like this. And so there's webbing between their toes, okay? But then there's also a mutation, which would be this version. This really bad version, y'all. My drawing is so bad. Is extra webbing. Okay, so now there's just a mutated little capybara that's born that has additional webbing. The webbing goes further up to the tip of their feet. Okay. And again, I'm making this up. And so if it's normal, if they have normal webbing, they are either big D, or big D, little d, right? They're either homozygous dominant or heterozygous, because this is a completely dominant trait. And what that means is there's only 2 options, big D or little d. Those are the alleles. Okay, so let me back up for a minute. When we talk about calculating allele frequencies, you know what the word frequency means, right? It's how common something is. Is it frequent? Is it infrequent? So how common, how often do we see it? The word allele is a word that a lot of AP Bio students feel like is an acquaintance. It's not your best friend. Okay, it's not a word that you know really well, but it's a word that you know about. You don't use it a lot, so you don't always remember what it is. Alleles are different forms of a gene. So if the gene is for webbed toes, there is a normal allele and there is an extra webbing allele. Okay, those are 2 different versions of the same gene. What's going to make them different is their sequence. So if you're looking at the A's, T's, C's, and G's, this one had a mutation, a change in the A's, T's, C's, and G's that led to a different protein that led to extra webbing. Okay, so that's what we mean by alleles, or just the different forms of a gene. All right, so allele frequencies is how frequently, how common is this allele versus how common is this allele. That's what we're going to be calculating with allele frequencies. All right, back to what I was saying. So if they have normal webbing, because this is a completely dominant trait, they're either big D, obviously, because they have two normal genes, or they're big D, little d, because they have a normal gene and an extra webbing gene, but the normal gene is dominant. So this, like this one, doesn't exist. For them to have extra webbing, they must be little d, little d. It's the only way to get extra webbing. Anything extra, anything else would not give any extra webbing. Okay, it would be normal. All right. So let's say that we went in 2010 and we surveyed this population of capybaras, and there were 2,000 total. Okay, so the whole population was 2,000 capybaras. And of those 2,000, 1,800 had normal webbing. Okay. Normal webbing, remember, is either big D or big D, little d. Okay. So now let's bring in Hardy-Weinberg. I mentioned up here that the dominant allele is P and the recessive allele is represented by Q. So if this is a dominant, this would be P, and this is a dominant, this would be P. So DD is the same as saying P times P, or P squared. Okay. So P squared is the same as big D. This would be P, this would be Q, so this would be PQ, but there's two of these because it can either be, I got the dominant or P from dad and the recessive or Q from mom, so that's one option, or I could have gotten the dominant from mom and the recessive from dad. So there are two options for me to get PQ, so I have to multiply it times 2. Okay, and then this is QQ. Okay, and you would multiply them times each other. So that is Q squared. Okay, now here's the problem. I know that 1,800 out of 2,000 is either big D or big D, little d. Okay, I don't know which is which. I can't tell by looking at a capybara. I can tell by his feet which one of these he is. But if he has normal webbing, I can't tell by looking at him if he's homozygous dominant or heterozygous. Heterozygous has two variables. So I cannot do anything with this 1,800 because I'm not going to be able to solve for two variables. But what I can do is I can use this 1,800 to help me figure out how many have extra webbing, and then that only has one variable Q. Okay, so subtracting the 1,800 from the 2,000, that gives me 200 that have the extra webbing. 200 out of 2,000 is going to be my little d, right? Because they actually have the extra webbing. That's a phenotype. I can see that physically on them, but I know because that's their phenotype, this has to be their genotype, okay? And because I know that's Q squared, when I calculate that, that is 0.1. So I know that Q squared is 0.1, okay? Q squared is not an allele, right? Allele is P and Q. So I need to get to Q. So if I know Q squared, how do I get to Q? Well, I just take the square root, right? So 0.1 square root is 0.316227766, and there's probably more numbers, but I am going to round that off to 0.32, okay? Now, there are two formulas that you need to know for Hardy-Weinberg. Well, you don't need to memorize them because they're on the formula sheet, but you need to know how to use them. The first is P plus Q is 1, okay? So if I know that P plus Q is 1 and I can plug Q into there, I can subtract Q from 1 and get P. And so P is going to be 0.68, okay? Because that's what I get when I subtract Q from 1, okay? The other formula is P squared plus 2PQ plus Q squared equals 1. Okay, so why does this work? Well, this works because there are only two options, big D and little d. Okay, big D is P, little d is Q. If there's only two options, when you add them together, they have to equal 100%, and 1 is 100%. So if I add all my big D's and all my little d's, I'm going to get 100% because there's no other option. Everybody either has a big D or a little d. Well, actually, all the capybaras have two, but all of the alleles are either big D or little d. All right, looking at it this way, this is basically saying that if I add up all the genotypes, right, homozygous dominant, heterozygous, homozygous recessive, then I'm going to get 1 as well, or 100%, because again, there's only those three options. So when I add the three together, they have to equal 100% because you can't, there's no other option to be anything else, okay? Anyway, all that to say, what we can do now that we know these numbers, you always want to make sure they equal 1, but they should because you used Q to get P, is we want to figure out, there's a couple different places that AP can ask you to go from here. So these are the allele frequencies, okay? I do want to graph it because the I can statement says I can calculate allele frequencies and show them graphically. So I can calculate them. Can you get P and Q? Okay. And then I can show them graphically. So I've created a graph. I went ahead and got it started just so I didn't waste your time building a graph. I've got allele frequency on the y-axis, the alleles on the x. It's just going to be a bar graph. Okay. And I know that my Q is at 32%. Let's do Q. All right, 10, 20, whoops, 10, 20, 30. So it's just over, ooh, I made that a little fatter than I meant to. Okay, just over 30%. And my P is just over 60%, okay? So, or actually it's not just over, it's almost 70% because it goes up to 68, okay? So I'm just going to fill that in. And those would be graphing my allele frequencies. So super easy to graph your allele frequencies. You just graph the percentages, okay? AP is rarely going to ask you to do this, but they might. So just be prepared. Okay, so it says I can calculate allele frequencies and show them graphically. Can you calculate P and Q and can you graph them? Okay. Then the next place AP might want you to go, and this is going to be the other two I can statements. I can explain how Hardy-Weinberg equilibrium is used to quantify evolution, and I can use evidence or data to determine whether or not a population is evolving. This is the first step in that, okay? One thing they may ask you is they may ask you the genotype frequencies. Okay, remember, and this is a key thing you need to know the difference with Hardy-Weinberg. Are they talking about allele? If they're talking about allele, we're talking about P and Q. Or are they talking about genotypes? Genotypes are big A, little a, little a, little a. Okay, so big A is P squared. P we said was 0.68. Okay, and we square that. That is 0.4624. So I'm going to round that to 0.46. Big A, little a is 2PQ. So I'm going to do 2 times 0.68 times 0.32. Okay, 2 times 0.68 times 0.32 is 0.4352, so 4. I round everything to 2 decimal places. It's generally what AP wants. They'll tell you if not. And then little a we have up here. Okay, we don't have to do that one again. That was Q squared, and that was 0.1. Okay, one way you can check your work is you can add those 3 together. And remember, the 3 of them together should equal 1. So 0.46 plus 0.44 plus 0.1, it equals 1. So you can always do that to check your work, make sure you didn't hit a button wrong on the calculator or whatever. So that's one thing they may ask you is they may give you this, 1,800 out of 2,000 have normal webbing. How many are heterozygote, or what's the frequency of the heterozygote? And so you would have to calculate Q and P, and then from there you would have to calculate 2PQ to say, okay, it's the frequency of 44%, okay, or 0.44. The other thing they may ask you is the number of individuals with each of these genotypes, okay? To calculate that, you take these frequencies and you multiply them times the whole population, okay? So we know that these two equaled 1,800. What we don't know is how many of the 1,800 are this and how many of the 1,800 are this, okay? We do know that these are 200. So we know that there's 200 of those, okay? Because we calculated that earlier. And you can double check that by doing the 0.1 times 2,000, and that gives you 200, okay? But what we want to calculate first is this one. So 0.46 times 2,000 is 920, okay? 0.44 times 2,000 is 880. Okay, and then again, if you wanted to do the 0.1 times 2,000, you can, or you can just get it because we already calculated it here. You can add these up, make sure they equal 2,000 or close because we did some rounding. They might be a little off. But that's another question they may ask you is they may say, 1,800 of 2,000 are normal webbing. How many of those are heterozygotes? And so you would have to calculate P and Q. You'd have to calculate the frequency for the heterozygote and then multiply it times the 2,000 to say, okay, of those 1,800, 880 of them are heterozygotes. Okay, so we know that a little above half are homozygous dominant, a little bit below half are heterozygote. Okay, so those are kind of the ways that they might use the Hardy-Weinberg equation on the AP exam, but they may take it a step further, especially if it's a free response. They're going to want to make you do a little bit more work. Free responses generally have multiple parts, especially the first 2. They may go down to like part F or G. The last The last ones are generally like A, B, C, maybe D, but usually they're a little bit shorter, okay? So what they may do is they may start you out with this information, okay? And that's all they would tell you. 1,800 out of 2,000 in 2010 had normal webbing, and you have got to figure out the rest of this, okay? Then they may take you down the road a little bit and say, you know, the scientists went back in 2050. I can remember what I had on the graph over there. And when they went back in 2050, what they found is the population had now increased to 4,500, which is good, right? The capybaras are doing good. The cute little guys are doing fine. But now when they check their toes, they find that 2,500 have extra webbing. And if you'll remember, extra webbing was our recessive trait. Okay, so this time they're giving you Q squared. Okay, they're starting you off with Q squared. And that's one of the key things with Hardy-Weinberg. You've got to know what they're giving you, and you've got to know where they want you to go as far as the P's and Q's. Okay, so what we can do is 2,500 divided by 4,500 equals Q squared. Okay, so you plug that into the calculator. That gives us 0.55555 forever. So we're going to round that to 0.56, and that is Q squared. Okay, from that I can take the square root to give me Q, and that is 0.75. Okay, because we know that P plus Q equals 1, we can plug Q in, subtract it from 1, that gives us P is 0.25. Okay, so now we have P and Q, we can do whatever they want us to do. What they might do, literally all they might give you, is this bit of information. In 2010, there were 2,000 capybaras and 1,800 had normal webbing. We went back in 2050, they're now 4,500, 2,500 have extra webbing. Did evolution happen? Literally, that might be all they give you. And you have got to figure out what to do from there, okay? So what you need to do is you need to calculate the allele and genotype frequencies for both sets, okay? So for the 2010 set and for the 2050 set, or 2050 set, okay? So we've got our allele frequencies now. Allele frequencies. And we're just going to make another table like we had a minute ago. So we're going to do our genotype frequencies. Okay, so we write out our genotypes. Genotype frequencies, number, and I'm going to go ahead and do number of individuals, and I'll tell you why in just a minute when I get there. But we're going to do the frequencies first, okay? So big A we know is P squared. We know that P was 0.25. When you square 0.25, it is 0.0625. Oops, 06. And I'm just going to round it to 2 decimal places because that's where I like to live, and that's generally where AP likes for you to live. Okay, big A little a is 2PQ, so we're going to do 2 times 0.25 times 0.75. And that is going to be, oops, 0.375, which we're going to round to 0.38. Okay. And then little a, we have from up here because we still remember it. And again, if you want to stop here and check your work, make sure that these add up to 100. 0.06 plus 0.38 plus 0.56 gives you 1. Okay, that's, again, checking to make sure you didn't hit anything wrong on the calculator or whatever. It takes a second, and it's a good use of your time on the AP exam because it only takes a second. All right, what we're going to do now is, in order to say these are evolving, now let me pause for a second here. You can probably guess evolution has occurred because your allele frequencies have changed, and they haven't changed from like 0.32 to 0.33. They went from 0.32 all the way up to 0.75, and from 0.68 down to 0.25. That's a pretty significant change, says my eyeballs. And so you can be pretty sure that evolution has occurred because those have changed. But what we cannot do with our eyeballs is we cannot say, as I just did, we cannot say that it is a significant change without the math, the statistics behind us to support that. So that's why we're doing this last step, is to show that yes, it looks like this is a change. And so yes, it looks like evolution is occurring. We're going to do some statistics to prove, to support that and say, yes, that is a big enough change for us to say it is a statistical change, okay? I also am going to graph the 2050, these numbers in just a second, just so you can compare the graphs. Okay, but we'll do that in a minute. All right, so what we're going to do now is we're going to take this 0.06 times our new population of 4,500. And that tells me that of these 4,500, 270 of them are homozygous dominant. 0.38 times 4,500 is 1,710. And then we knew that we had 2,500. I will say this, because we rounded, these numbers are not exact. For example, if you'll remember, this was 0.375. If we did 0.375 times 4,500, it is 1,687.5 capybaras, so 1,688. What AP is going to do, what AP does, is they know that you may round differently than they do. Now, they're going to give you a specific place in the final answer that they want. Like they might want the final answer to be in hundredths place or tenths place, or the nearest whole number, but they're not going to tell you how far to go along the way. So you might have rounded to 2 places here like I did, or you may have left it 0.375, in which case you would have gotten 1688 here. AP knows that, and so if the question was how many individuals are heterozygote, they would accept anything from probably like 1685 up to 1710. They're going to give you a range because they know that you might round a little bit different along the way, and so your final answer might be a little different. So just be relaxed about that. Don't freak out thinking, oh my gosh, what if I don't get the exact number? Because if there's any rounding along the way, the final answer is going to be a range, and you'll be fine. As long as you do it correctly, you'll be within that range. Okay. So that's what, if you add those up, they're not going to equal 4,500 because we've rounded. So just know that, and that's fine. You could have done 0.56, times 4,500, and this would have been 2,520. So instead of relying on the number they gave you, you could have applied this. Again, because we rounded, it is a little bit different, okay? All right, so those are our numbers. What we want to do now, and I'm going to do it in green just so it shows up a little bit better, is we are going to do a, well, let me write this. So I'm not trying to talk and write at the same time. We're going to do a chi-square, okay? Chi, that was real Southern. Chi-square. We're going to do a chi-square. So chi-square is going to allow us to say, yes, those numbers are significantly different, or no, they're not. Okay, so our null hypothesis is that the population is in Hardy-Weinberg equilibrium, which means that these percentages down here still apply. Okay, and no evolution has taken place. So the first thing I need to do is I need to create, whoopsies, not that. I need to create my chi-square table. Okay, it's going to have my genotypes on it. So homozygous dominant, heterozygous, homozygous recessive. It's going to have a column for observed, a column for expected, and then a column for me to calculate my chi-square. Okay. So where do we get these numbers from? All right, the observed is what I'm observing in 2050. So it's my 2050 number of individuals. Okay, I'm not going to do frequencies, because chi-square doesn't pick up differences very easily in frequencies, okay, in decimals, I should say, in things less than 1, in tiny numbers. We're going to be able to see the difference more clearly here. So I'm going to put my 2050 numbers here. So if you have to do a chi-square, you're going to have to do this step. Okay, 1710 and 2500. And then my expected, let's think about what this is. My expected is if my null were true. If my null were true, this population is in Hardy-Weinberg equilibrium, and there's been no evolution, which means these percentages, let me fold this in half. These percentages from 2010 still apply, okay? If these percentages from 2010 still apply to my new population of 4,500, what I need to do is I need to multiply these percentages times 4,500. I cannot use these numbers because these are from my 2000 and my population has grown. So what I'm going to do is I'm going to take my 0.46 and I'm going to multiply it times 4,500. And so if we were in Hardy-Weinberg equilibrium and if these percentages still applied, 46% of 4,500 is 2,070. So that's how many I would have expected to see be homozygous dominant. If the 0.44 applied, then it would be 1980, okay? Because 44% of 4,500 is 1,980. And then 0.1% of 4,500, I don't need a calculator for that. I could do that in my head, is 450, okay? And so those are my expected values because they're my percentages from 2010 times my new population. Okay, that's why we couldn't use the numbers that we got on this table because they were for 2000. I need it to be for 4,500. Okay, so now that we have observed and expected, you know what to do, right? You do 270 minus 2070. That's not what I did. I did divided by 270 minus 2070. Square it. That's a huge number. Divided by 2070. 1565.22. I'm just going to round everything to 2 decimal places. 1710 minus 1980, square that, divided by 1980 is 36.82. Okay. And then 2500 minus 450, square it, that's a big number, divided by 450 is 9,338.89. That's probably the biggest number I've ever seen in chi-square. 1565.22 plus 36.82 plus 9338.89 is 10,940.93. That's my chi-square value. Guys, that's huge. I'm sorry, I have a tickle in my throat. All right, a p-value of 0.05. Degrees of freedom. This is hotly debated. Not really, but I don't like it. It's hotly debated in my brain. I want degrees of freedom to be 2 because we have 3 groups, right? Degrees of freedom is the number of groups minus 1. Sorry. All right, so 3 minus 1 is 2. But that's not how it works in Hardy-Weinberg equilibrium. In Hardy-Weinberg equilibrium, you're actually, yes, you're doing your calculations with these, but you're looking at your 2 alleles as your groups. So actually we have 2 groups minus 1 is degrees of freedom 1. So that's just something you need to remember with Hardy-Weinberg equilibrium, that your degrees of freedom is 1 when you really think it should be 2. All right, and then our critical value comes off of the chi-square table, which you do not have to memorize. It's on the formula sheet. You do have to use your p-value and your degrees of freedom to find it, and that's 3.84. Now, when your chi-square is larger than your critical, and good grief, it is larger than our critical, we are going to reject the null. And what did our null say? That we were in Hardy-Weinberg equilibrium and there was no evolution. We are rejecting that. So that is our evidence, our statistical evidence that evolution has occurred. Okay, so let's go back and look at our I can statements. I can calculate the allele frequencies. That's P and Q. Okay, can you, given the data, if they give you whatever, do you know what they're giving you and do you know how to get to P and Q from there? That's the key. Okay, that's the key for that I can statement. And show them graphically, which I forgot to go back and show you your 2050 numbers graphically. All right, P dropped down to 25. Okay, and Q jumped up to 75. Hopefully I'm eyeballing that correctly, getting into the right place. Okay, so again, if they want you to show them graphically, that's all you're going to do. You can look at these graphs and see that's a big change, right? I don't know if it'll bleed. I don't know if you can see it through. Can you see it's kind of up to there? Y'all can't really see. I can see it better with my eyeballs. But anyway, so dominant trait dropped pretty drastically. Recessive trait increased pretty drastically. But again, that's not enough to say there's a change. I mean, you can say there's a change, but you've got to have the statistics to say that it's a statistical change. It's a big enough change to say, yep, that's a change. So that's calculating allele frequencies and showing them graphically. I can explain how Hardy-Weinberg equilibrium is used to quantify evolution. That just means to assign a number to, ooh, crazy here, assign a number to or calculate count. And so that's what we did here. We showed that our allele frequencies changed, right, from 0.32 and 0.68 to 0.75 and 0.25. Which shows that evolution is occurring. Again, we have to do the chi-square to prove that that's a big enough change to say it is a true change. But when it's that big, you can be pretty sure even before you do the statistics that it is a true change and evolution is occurring. And then I can use evidence or data to determine whether or not a population is evolving. So again, we've used our data from our capybaras from 2010 and 2050. The evidence that was collected, right? We're using that data to show that there was a change in allele frequencies There was a change in the expected numbers of each genotype, and so therefore evolution is occurring. So I hope that helps. If you still have questions about Hardy-Weinberg, how does it show evolution? How do you do allele frequencies? If you're continually starting in the wrong place, like you think you are supposed to be starting with Q, but actually you should be starting with Q squared, things like that, let me know and we'll practice me just giving you some examples and you telling me, is that Q, is that Q squared, is that, you know, P, what am I giving you? So that you can, because that's the key. If you don't know what they're giving you, you're going to start the problem wrong and then the whole thing's going to be wrong. So you just want to make sure that you're starting off correctly. You want to know what they're giving you, but also what do they want from you? Where are you going? Okay. And that's that. So if you still have questions, need extra help, let me know and we'll talk about it. Thanks, guys.